Hello everyone, my name is Kaled Jumah and I will take you today through the Galaxy training material on detecting structure variants from exome sequencing for diagnostic genetic disease. And in this tutorial, we will talk about the steps you need to take in order to detect structural variants from exome sequencing data, which is different from the whole genome sequencing data, as we are targeting the regions responsible on the translating into proteins. And the reason we take this approach is the cost efficient of sequencing the regions that responsible. We know that responsible on protein translation, and this is for specification. And we reduce the cost of sequencing the whole region of the DNA. And in this tutorial, we are detecting structural variants from a trio family, father, mother, and son, where the patient or the son has the structural variant and or the disease. And in this way, in this study, we are locating if these structural variants caused by de novo mutations or loss of heterozygosity. And in this tutorial, we will get through two different approaches. So one approach is if you start with the sequence data, and those are the FASTQ files, and then you go through the quality control, read mapping, and the variant calling, and then the variant calling annotation. And so the two pathways is we can start from the raw data, or we can start from the map read, and this depends on how progressed you are with the previous tutorial. So if you followed the map read mapping, or you go through the deployed previous tutorial in this learning path, so if you have the deployed system training, you can scroll over the tutorial for the mapping and then proceed with the variant calling step. But we will take a step-by-step for this training. So for the data preparation is when you start after the read sequencing from the map. After the read sequencing from the sequencer, the Illumina, you will get a short reads, and those reads are scattered around for the whole reference genome, or for the whole, so there would be reference genome, and those will be as part of our sample genome. And in order to build the whole, to know the location or these each short reads where it belong to, we need to map them and reference them. So starting by getting the data, so the data will be saved on the Zenodo, and you can upload this data into Galaxy through different approach. You just can copy and paste those links into the Galaxy through the upload and pass fetch. You can see that here, and then it will be uploaded into the Galaxy, and you can also upload the data if they are stored or saved in your local computer through the upload, and then you go choose local file and etc. So but if you have followed this previous tutorial, then yes, you can proceed. The next thing, if you already familiar yourself with the mapping and the quality control steps, you can start from the map read and you go proceed with the variant calling detection. And also it's important to know, is good to annotate the data and give them a meaningful names, as well as if you want to work with the built-in reference genome, you can use the Galaxy ones, or Galaxy already have a multiple reference genome stored. While if you want to have a specific version or specific type of reference, you can install that and upload that to Galaxy, and the tool will accept that. And for the quality control, so after you got the sequence read, so you will have them how the FASTQ file works. Then you will have the title of the sequence, and then you will have the sequence read, and then this is the quality score, and before working, like once you receive data, it's always a good approach to check the quality of the sequence read and to see if you need to fix them, prepare them, work with them. As in, we do the quality control, and the quality control give us the test and the checks for the sequence we have, and it depends on the score. So it has something called the quality score, or the quality score, and this quality score with a maximum of a 60, I think. And then if it's the higher the quality, the better your reads are, and the lower they are, the bad, which means the precision of the sequence. How was the sequence precision that this nucleotide is have been called correctly and in the correct position. And for here, the GC content, so it's failing for the GC content, but in general, the GC content resembles that if you have samples are contaminated or not. And once you do the quality control, some data, so once you're done with the quality control, you proceed, and then when we have, in general, we have a good data. It's not required to be trimmed as they are good quality, but if your data has a bad quality, you can say it, or for example, you have like reads here in the red, then you should consider trimming these sequences. And the quality control also give us an idea on when to discard the data. So if your whole sequence you have is it in with the region, means that it is good idea to skip, or like not proceed with this data and ask different data due to the poor quality. So it will save your time in the future. And then we go with the mapping. So after we have the short reads, we need to align them to the reference genome in order to identify where they belong to what chromosome. So the BAM file. So we will use the pairwise BWA aligner, and that aligner align those short reads of the reference genome, and it tell you the information of that. So it can show you the chromosome, and it also tell you the gene name, the start, the end position, and those short read and other information. So once we have done with the mapping step, then if you follow the tutorial, first you map the read of the son, and then you map the read for the father, and then the parents. And once you get that, you want to ensure that you are keeping the data that have been mapped properly. So we are working to detect the structural variants, and those are a bit tricky to detect. So we want to make sure that our data has been mapped properly before proceeding with the analysis. So we will use the SAM tools view. So the SAM tools view work into filter the data. So we want to filter the data that hasn't properly mapped. So we will use, if you go for the SAM tools view, so the SAM tools view, and yes, I have run the workflow before, so I will show you at the end how to run the workflow from the training, but you can proceed with running tool by tool. So we want to filter the data. So the SAM tools view filter, it is part of the SAM tools, and we want to filter on a selected reads, and then we select those parameters as the way we are filtering the configure filter. So we are filtering the data that if the read is unmapped, or the reverse read is unmapped, so we ignore, we filter them from the results. So we ensure that all of our results or all of our sequence had been mapped properly. And then there's another approach of mapping. So there's another stint of mapping. So what we have used now is we just filter based on if the read is unmapped and the mate is not mapped, but there's a stronger filter approach which is we call it the proper pair, and it filter unmapped reads, and this is like a further filtration. It's selecting the best filter is depend on your research criteria. So if you filter the data too much, you might miss some of the data, for example, copy number variance, but it's the selecting what is the filter is you should consider in your analysis. And then once we're done with that, the provias. So what I call the proper filtration, which is read is mapped in a proper pair. Then we remove the duplication. So what we use SAM tools. So remove duplication is also part of the SAM tools. So what does the remove duplication does is when you have the aligned sequence, and some of those sequence is sequenced during the PCR, they will be duplicated, or they will be duplicated more than the other sequences. So we use remove duplication to get rid of this bias. So it will take off the sequences that have been duplicated, or they have been duplicated more compared to the other sequences, and proceeding with the variant calling. So we will use the free bias. And the free bias is a Bayesian genetic variant detector, and we use it for detecting SNPs, multinucleotide polymorphism, and they work with the short and long read variants. So after what are the inputs? So the input would be the outputs of the map reads after the filtration. And then we use the reference genome, go for the training material here. Let's go for the Galaxy. And so this is the free bias. So what is our input? So we will use the reference genome, which is locally cached, but in your case, you can change that from the history if you have installed it from there. And then we want to get the VCF file. And when we say it's a variant calling format, and it's made by the GA4GH, and you put the BAM file for the remove, like the filter data for the trial family, and you do some of you manipulate the parameters. So in our study, it's a diploid calling, and we are detecting the variant from a diploid family, and then you run the tool. The output of the free bias is the VCF file. And the VCF file, so this one, it'd be the header, and in the header it should also be called later on. So yep, so this is a different, it should be called here. So we will talk about them later. So the hashed line is the identifier of the file. It tell you about the steps that have been done to analyze the data, what type of the mapper, what type of the tool have used to detect the variant analysis, and then you will have the header, the icon telling you later on what each column represent. So you will know about the chromosome, the position of the variant, and what is the reference and the alternative nucleotide, and then it gives you the quality. So the quality is based on it's the log value of it's called the Fred score, if I remember correctly, and the higher is better. So if it's 10, means that the accuracy is 90%, and if it's a 20, it's a 99.9%, and etc. And they give you another information about the variants, such as where it's located, the type of the variants, and annotation data. So they can be expanded later on. And then you got the format. So you have the genotype deployed, which is if it's 0, 0, then there's no reference, the changes compared to the reference. While it's 1, it's like there's a difference in your the heterozygous, it is in the heterozygous. So we are human, built on 2 strands of DNA. So one come from father, like a paternal, one is maternal, and we check the difference. We check the difference in that allele with the reference genome. So it's 0, 0. Then there's no both alleles are similar to reference genome, while 0, 1 we have one allele different to the reference genome, and etc. And this also take you through the tutorial. It tells you that the combinations or the components of the VCF file. And then we have the post-processing free bias call, and this is also for the standardizing of these variants in a way that we have variants, or the variants that we have detecting. For example, we have a deletion here in this reference genome and the alteration sequence, and what we have is we want to call that alteration in a way that is a standard by for one of it trim the extra sequences or trim the extra nucleotides, and also only keep the minimum sequence to resemble that alteration, and also move that into the closest position. And why do we do that is if we follow how it's called normally, we will end up with multiple variants for the same. We will end up with multiple variants for the same allele, or multiple locations or multiple forms for the same allele. So we do that in order to standardize the alleles or the variants for the sequence we have, to compare them later on. And if you prefer to do that, so go for this training material, like this documentation or this paper telling you about how this works and how what is the important for the normalization. And then we go for the annotation. So what is annotation? After we work with the normalization using the BCF tools, we ran down into the galaxy, and now we have them in a standard format. So you can see here we have the references, the cytosine, and then this would be addition to the sequence we have. But at the end, we don't really know what is that effect. So what does that affect? So we have these variants, and we don't know if they are associated with disease or not associated with disease. And here comes the annotation. The annotation means that we add information into the data we have. And so we will use the snip effect, and the snip effect will add so the snip effect will to proceed. We do the annotation step to identify the variants we located. Are they pathogenic or not? As some of, or I would assume researchers or unexperienced researchers would assume that if we detect a variant deletion on the coding genome, or which is the exome, we would assume that is a pathogenic. However, not all pathogenics, or not all of these variants are pathogenic. Some of them are benign. Even if they change the protein structure, there won't be any change in the function of that protein or the phenotype. So they would be responsible of the variation of the phenotypes among species or within the same species. And so in the annotation, the term is prioritized. So we prioritize those variants. We know the variants associated with diseases or the changes or the pathogen changes of the protein. So we detect those, and then because we have also a diploid system, we have a family, the father, the mother. So if those variants have exist in the father and the mother, each one of them or both of them, and this variant does not hold a pathogenic effect, this also can be waived. And for the gene annotation, calling and manipulation, we will use Gemini. The Gemini is a database like it helps for annotation and extracting the data. Uh, if you extract that, so you go for this link for the structure of Gemini, which is I all strongly recommend. To work with Gemini, we can directly upload the output of the normalization for the VCF directly. We will use the snip effect. So the snip effect will annotate the data and also will create an output file to be used and manipulate for the for the Gemini. And to before we run the data for the snip effect, we need to create a PED file, and this resembles the samples we have, and that's also important. If you follow the training, it's when you do the mapping, it is an important step to make sure that you name the sample based on the same name you would use for the PED file. So if the name is father, mother, and proband, and then you do the parental ID, maternal ID, sex and phenotype, you proceed with the snip effect. So you download the annotation for that, and the output of running this, you will get the HTML of the snip effect, and it will annotate, and it will give you a nice output or nice feedback on what the data you have. So you will get the chromosomes, the length of each chromosome, and each variance within that chromosome. You can see a lot of variance here in here, and it's divide your variants into different types. So you have deletions, single nucleotide polymorphism, multiple nucleotide polymorphism, and then their order and many more information. So I would recommend you, and it tell you that we have and what type of the alteration we have, and yeah. So basically the positions and additional information. When you proceed with the training, so after detecting the snip effect, we want to do the Gemini, and the Gemini help us in first of all, it will store the variants, and it like one of all, Gemini is part of the SQL and SQL search. So I would recommend you to have like at least skill building skills on how to search an SQL database. If not, it will be mentioned later on the tutorial on how the link on how to work with that. So we need to upload. First of all, we need to upload the data into Gemini. So we will use the Gemini load. So the output of Gemini load is the Gemini database. And to check that is you go for the create a tabular file to get multiple files, so a different format. So to work with that. So but with the SQL. So this is the code, the tabular of the data we have here. It's called the chromosome, the start, the reference, alteration, where is the gene located, and then for also the clean the output of the clean var significance. So some of those are like not, they have a non effect, or they no data, and then you got benign, likely benign, pathogen, likely pathogen. So to determine the effect of that, and also it can be amended. You can amend that later on, and then you include many information. So you use the Gemini load. So we load the output of the annotated VCF file into the Gemini database, and here is the Gemini suite tool. So So Gemini has a different tools, so it can load, it can annotate, it can amend. So if you want to amend the Gemini, and then you can do the Gemini annotation and database information. The query is for experienced people with the SQL. However, we will discuss that later. And then for the variant detection, and this is a basic information on Gemini, how it works, and for the detection. For this tutorial, proceeding with the Gemini in order to for the inheritance pattern. So what they would raise a question is, what is the inheritance pattern for the phenotype we have in the study? So is it autosomal recessive? Is it it's like an autosomal dominant? What do we mean by that? So once we have the variant, the deletion, so the deletion deleted the like a nucleotide or like an allele which was a dormant, and deleting that, it masked the it masked, or it removed the masking of the recessive mutation. So this is a list of the type of the mutation, and what we have. So this is a question to answer, and if you don't have the information on, or the basic the background on those, so the autosomal recessive, which is a dormant mutation, cannot be shown unless it's both of the strands are, the alleles are dormant. The dominant is, it can be seen either in the one of the strands, like one of the parents, or one of the strands, and this is like an X-linked, or and it's a de novo or the loss of heterozygosity. Uh, in our case, it's if it's the variant is not seen in, is only seen in the son, while not be seen in the parents, that can be a de novo. Or if it's she's seen in one of the paternals, for the parents, it can be led to the loss of heterozygosity. And to detect that, we will use the Gemini inheritance pattern. And for the Gemini inheritance pattern, it will detect those regions uh, based on the assumption. So we will use the Gemini load, so the database we have created in the Gemini, and then we will use the autosomal recessive. So we are suspecting that it's an autosomal recessive, and then with the impact severity, we use low, and that's for we will extract the data uh, for like we will not extract, we will ignore the data that have the impact on protein function. So we will only look into the data that they have a high impact on the protein, and then with the following of the parameters, so include hits less than like the inheritance pattern, we use no, and follow with the training, will give us an output of the it give us an output on the variants. Sorry, will give us an output on the variants that they have a strong effect on protein in the protein coding, and the for the not autosomal recessive, which is the inheritance pattern. So we would know the variants that would be associated with the de novo or with the loss of heterozygosity from our sample. And at the end, the conclusion is it's not really hard to detect the variants from child disease in the deployed system. It's only part of the steps you should follow. Gemini is a strong tool for the annotation and annotation manipulation. It is good to expand the horizon with what Gemini can provide. I strongly recommend follow other training materials. There will be another training material in mentioned the advanced for using Gemini in more precise searches. I got mixed up with this training and the next one, so you can find that in the learning path. I will just show you here. So the variant analysis, and that will be identification of somatic variants compared to normal. And in this training, there will be an accurate how to use the Gemini for the query annotation, and then how to use annotate and do annotation from different resources. I strongly recommend you to follow this training or do this training after this one. And at the end, I really appreciate your time and joining this training material. It's good to follow that in your base and have a good day.